After download the CellPie zip file, you can unzip it to any folder. You will find a logo folder, a CellPie.exe, and agreement text file. Double-click the .exe file to start the program. To get rid of the unnecessary processing outside of the interesting area, we provide a sub-image crop in the first step. Load the original image. Draw a rectangle RY on the image. You can adjust the RY only on the interesting area to get the sub-image. Finish. The processing interface will appear. In this interface, there are 3 regions, including the processing image, the plots for pie structure, and the operation region. In the operation region, there are 3 parts: image information, segmentation, and distribution evaluation. In the image information, there are basic image informations, including the parse, the resolution, and the pixel unit. You can define it later. The first step for operation is to segment the cell pixels. We provide a threshold methods on different channels. Here we choose the blue channel, adjust the threshold. All the segmented cell pixels will be labeled in green. You can disable it to compare. Here shows the pixel numbers of the segmented cells and the area. The area is related to the defined pixel unit size. We also provide a ROI function here to add or subtract some regions using its cell or not. For example, we can get rid of this region by subtract, or draw ROI to add them. After segmentation, we have to define a centroid to see the pie structure. Here are 3 ways, including the cell centroid, ROI centroid, and the manual centroid. The cell centroid is automatically calculated according to the segmented cells, and the ROI cell centroid is calculated by the segmented cells in a ROI region. For example, if we only want to see the centroid in this region, here is the centroid calculated. Also, we can define it by manually. After define the centroid, we can see the pie structure, which describes the distribution around the centroid in each direction and in different region. When click any of the block, you can see that distribution along that special directions. You can adjust the parameters of the pie structure. Also can change the maximum radiance. We provide 4 data types, including pixels, pixel multi-intensity, pixel per area, and pixel intensity per area. Pixels means how many pixels in that region. And the second is how many pixel multiplied by their intensities. And for the last 2, they will divide by the area size. With the pie structure, we also have a look of its 3D distribution by clicking the 3D view. It will generate a 3D pie view. You can use the rotate to see how they distributed.